All right, so how do we apply these markers? Particularly, as I said, the SSRs and the SNPs are the ones that people are using the most. How can we use them in rust control? I mean, that's what we're about, isn't it? We want to control rust. We don't want to sit in a lab and just generate results and publish papers in good journals. That's a nice thing to do. What we want to do is we want to help farmers control rust. So how do we use molecular genetics to control rust? First of all, they can be used in plant improvement. So they can be used to actually locate genes of interest. This is what we call a genetic mapping. We can develop markers linked to those resistance genes that allow marker-assisted selection. So in other words, selecting for the marker, if it's a really good marker, we don't even have to do a rust test. If the marker's there, the gene's there. Cloning of resistance genes, transformation to introduce cloned genes. They're 4 examples of the way we can apply molecular biology to improve rust resistance in wheat varieties. When it comes to the rust pathogens, they help us understand genetic variations. So we've already said we can have 2 pathotypes, 2 isolates of a pathotype, and their genotype is completely different. All right? And I'll give you an example of this as well, but that tells us that we're dealing with different genetic entities, and if we find the same pathotype in 2 separate places that are the same genotype, then it implies we've had migration from one region to the other. So it's telling us a bit about the epidemiology of the pathogens. And in pathogen diagnostics, which is, I suppose, the example I've just given you, where we can use DNA markers to help us with doing rapid diagnosis. We can now take a single pustule off wheat of stem rust from East Africa, and within 24 hours or 48 hours, 48 hours, we can confidently say this isolate does not belong to the UG99 lineage. If we do that in the greenhouse, it's going to take us 2 to 3 weeks. So this is where the application in DNA diagnostics is getting very, very good. All right, so I'm going to give you an example of how we go about using markers for mapping. All right, so you would have seen in some of the talks how we've said this particular rust resistance gene sits on this chromosome, chromosome 2BS or 3BS or 2DL or 7DS or whatever. How do we go about putting a rust resistance gene on a chromosome? In the past, we used a process called cytogenetics using aneuploids, monosomic analysis. Now we can use molecular markers. All right, so let's go back to the example I've shown you a few times in previous presentation, where we've used the gene-for-gene hypothesis and an array of pathotypes testing wheat germplasm that we didn't know the resistance status of, and we found one line that was resistant to all of our pathotypes. This is what I've been calling this genotype, wheat genotype unknown Y. It gives a low infection type, a naught infection type, to all 4 pathotypes. And on that basis, the other day, I said to you that we would probably say that this genotype carries either LR9 or LR19 or another resistance gene, a new one that we haven't found before. All right? How do we go about showing that it's new? How do we go about mapping it if it is new? So I did tell you what we do to determine whether it's new or not. We actually would cross, let me just go back one. To determine whether it's LR9 or not, we would cross this wheat variety with transfer, right? And then we would look at the progeny, and if there were no susceptible segregates in the progeny from that cross, we would conclude that it's probably the same gene, probably LR9. All right, I did tell you this the other day. I said if it is LR9, it's the equivalent of a cross of big A times big A, big A. And the only genotype you can get out of that cross is big A, which of course will be resistant. So if we intercross this with transfer and then look at the F2 and F3 progeny, and there are no susceptibles amongst those progeny, we would conclude that this gene is in fact LR9. If we did the same thing with Agatha, we would conclude that it's probably LR19. But if we get susceptibles in those crosses, then we say it's not LR9, it's not LR19, it's probably something new. And of course, if it's new, our breeders want that. They want that gene and they want to be able to breed with it. So what do we do to actually do the next step in the genetic characterisation? We have to do the mapping.